as you can see from my first slide, so we are a group basically working at the interfaces of organic synthesis and supramolecular chemistry, basically with an aim to make functional materials, basically functional organic materials. And most of the time, one of the component in our materials is like a kind of chromophoric system or a kind of biconjugated molecule, so that we give an optical and electronic functionality to these materials. Okay, so today's talk, I will be talking to about one class of material which is called supramolecular polymers, which is a kind of emerging material with fascinating properties or functions. But considering the broad audience, I will try to stick to the concepts rather than going into the details of the research topic. And as the title says, it's about microstructure control in supramolecular polymers. So although these kind of materials are used in various fields and so on, but one of the problems we are having is we don't have the structural or functional complexity what we would like to have for exploring its full potential. For example, if you look into the, if you take more than one monomers, we can make multi-component supramolecular polymers, but there is no way that we can control its sequence. But it is completely different than if you compare to the natural biological biopolymers, like if you take nucleic acids or proteins, we know that the sequence controls plays an important role in biology. Okay, and it determines most of the functions of these biopolymers. Okay, the question is, can we have some kind of control on the sequence of this kind of multi-component system so that we can address many more interesting functions out of this class of molecules? That is basically the title says, the microstructure control on supramolecular polymers. Okay. So I also thought it is very apt to talk about the supramolecular polymers because this year is kind of 100 years of macromolecular chemistry. So it was almost like 100 years back in 1920, Staudinger came up with the kind of classical paper of this polymerization, where he claimed that the polymers are like linked by monomers, linked by covalent bonding between these monomers. Okay, so it was at the early stages, many people were not ready to accept it. There were even opposition from famous organic colleagues and everyone. But it took almost some time to convince everyone that indeed polymers are linked by kind of covalent bonding between the monomers. Okay, and so it is almost like 100 years, and even ACS had planned a special session on this kind of next level of what should we do in the next century in the spring meeting. Okay, so I thought it is very appropriate that we talk about something similar, another class of yeah.
Subi, there is window coming up on your screen. Please move away, window away from. So is it like any background thing is opening, or I think now it's fine.
I thought you put it.
No.
I was trying to remove it.
Okay. Let me close few things if anything is interfering.
See, I was trying to.
Maybe just try to do it full screen now if it works. Yeah, so.
It's there.
Can you reshare the screen? Sorry for this interruption, but audiences are finding inconvenient to see your screen. So.
Yeah, I'm surprised.
Reshare, please. Put it on full screen now. Yeah, it's better actually. Nothing coming.
Okay, perfect.
Great. Yeah. Sorry. Please carry on.
Yeah. As I mentioned, so I will be talking about another class of material, which is very similar to the classical polymers, but we call it as a kind of supramolecular polymers. So the only difference is in structural wise, it's very pretty similar to the natural polymers, classical organic polymers or covalent polymers. But the only difference here is the monomers are linked together by very weak interactions in this particular case. It's not unlike the covalent bonds, they are linked by very weak non-covalent interactions. So as a result, one can imagine that these monomers are always in a kind of equilibrium. So this is a kind of video or to just demonstrate that how it is work. It is not a real video, it's just a kind of animation for a molecule which is shown below. It's a kind of benzene tricarboxamate, which has 3 hydrogen bonds at the periphery. So you can imagine that once you put this molecule into solution, they can come together through these 3 hydrogen bonding, a kind of lateral fraction. And also with pi-pi stacking and hydrogen bonding, they can form large one-dimensional structures, which is very similar to classical polymers. So the only difference is here that here, since the monomers are linked linked together by the very weak bonds. So they are always kind of, we can expect like kind of dynamic or reversible properties from this kind of materials. Okay, so the monomers will be always in a kind of exchange between the backbone as well as surroundings. So this field we should thank the pioneers like Taguso, Ada, and Bert Meyer, who really introduced this concept of supramolecular polymers probably 2 decades back. And you can see that large amount of research has happened in this field during the last 2 decades. And so the next challenges in this field is considered as, can we have a perfect control on its structure? Or in other words, can we have a kind of precision supramolecular polymers, or with the controlled degree of polymerization, that means controlled length and controlled dispersity. And another aspect is, can we have multi-component? Like I mentioned that talk towards the end of my talk, I'll be talking more about this aspect. Can we control the sequence of monomers in this kind of polymers? Okay. And finally, another challenge people have identified is, most of the polymers, if you look into it, unlike the biological systems, they are kind of at equilibrium. So one of the challenges to, can we go out of equilibrium to make non-equilibrium supramolecular polymers? That way we can bring very interesting functions. So these are considered as the new challenges in this field in order to completely exploit its potential. Okay, so if you look into the one more slide before I go to our research. So this is a kind of energy landscape of this supramolecular self-assembly process or supramolecular polymerization process. This is, again, you can see that to help you, there is a video available of a porphyrin monomer. So you can see that if you put it into the solution, it has again a porphyrin monomer with 4 hydrogen bonding at the periphery. It will come and form this kind of one-dimensional structures. And as I told, these non-covalent interactions, since they are very weak, they can easily form and break without much actuation barriers. And as a result, most of the synthetic supramolecular polymers, once you consider, they will end up in this kind of thermodynamic equilibrium, because it's easy to reshuffle and they will find the thermodynamic equilibrium very easily. So most of these systems, once you synthesize, they will be kind of stable over a period of time. And this is true because of there is a fast dynamics involved in the system because of the weak interactions. But consider the fact that if the dynamics is pretty small, sometimes it can get trapped into kind of excited states like high energy states like metastable state or non-equilibrium, kinetically trapped state. And with time, if you apply enough energy, they will relax back to this thermodynamic equilibrium. This is perfectly fine. But if you look into the nature, as I told, there are many interesting examples available in nature of similar to the supramolecular polymers. So this is a video, again, available in YouTube. It is about a kind of microtubulin filamentous proteins. Okay, so this is also kind of supramolecular polymer where individual protein monomers are coming together and arranging and forming a kind of extended one-dimensional assembly. But if you look into, there is a key difference between these two structures. On the left, it is mostly formed under thermodynamic conditions, but in this right one, it is more like a kind of out of equilibrium conditions. What I mean is, on the left side, it's a kind of energetically downhill process. The formation of the material is driven by gain in free energy. Okay, that's why the left side, most of the synthetic system works. But if you look into the right side, you need a continuous supply of energy for the molecule to remain in this functional state. We call it a kind of non-equilibrium supramolecular polymerization and so on. And this is very important in biology to control many of its functions. If you look into it, either signal transduction or cell division, everything, this motility, anything. So this kind of out of equilibrium supramolecular polymerization is a kind of key concept in biology. So nowadays, people are now, what synthetic chemists are trying to do is trying to move away from this classical kind of thermodynamically controlled supramolecular polymerization to kind of non-equilibrium supramolecular polymerization. So this is again another review written by the experts in this field, and they clearly call for we should move away from kind of this blue regime, which is a kind of equilibrium process, and we should move to this red regime to extract interesting functions out of this material. And another aspect we should think about is high complexity, because so far we have been dealing with very simple system with one kind of monomers. Can we go to multi-component monomers and make even more complex systems and so on? So these were the few challenges people are identified and considered as the grand challenges in this field to really extract more functions from these materials in the future. Okay, so these are the basic one aspect of our research, and with this challenges in mind, last 2 to 3 years we have, or 4 years, we have been trying to address various aspects of this. Okay, as I mentioned, one of the first attempt was to do precision supramolecular polymerization. That means make these structures with controlled length and dispersity. So something which is we call it as a kind of living supramolecular polymers. So I should mention that most of this research field of supramolecular polymers gained a lot of information, got a lot of information by comparison with these classical organic polymers. And as you go along the talk, you will understand that most of the principles are borrowed from classical covalent polymerization. Okay, and also all of you know maybe heard about this term called living polymers. That means it's a kind of chain growth polymers where there is no termination end. So as a result, what we can do is you can keep on adding monomers to these polymers. Okay, so then it will start growing at the living ends. So that's why we can control that's the way in conventional polymers people have controlled is size as well as the narrow dispersity. Okay. So the question is, can we have similar kind of strategies in living supramolecular polymers so that we can have a kind of control size and narrow dispersity, thereby making kind of precision supramolecular polymers. So the another aspect is, as I mentioned, can we go from this kind of equilibrium polymers to kind of non-equilibrium polymers? And that way we can make structures which are kind of transient in nature, similar to the nature. And then what the beauty is that this is one way to, although we are not able to mimic the biological system, at least we can make materials which has a definite lifetime, which can be predefined. What I mean is, so there is a functional state which is active for a while, and the lifetime of the state we can predict in advance by controlling the amount of fuel in the process system. So this is a kind of new kind of transient materials which are coming up, which will have interesting programmable functions. This is another aspect. And the next level of complexity I mentioned is very recently we started working on this kind of multi-component supramolecular polymerization, where we move away from one component system to the wish is that multi-component system, but at least go one step further to make two-component system. And can we control this sequence in such a way that we can make predictable microstructures in this kind of polymers? And some of the early results are coming out from our group. So the whole idea is that we should be able to make structures where the sequence, like in this case, is a red and green monomer. We tell the monomers where exactly it should come and organize. It's quite challenging. There are many interesting aspects to it. So these are and most of these cases, we are still at the early stages of understanding, and it requires many more years of research before we have a control similar to nature or most of the biological systems. Okay, so as I told, now let's talk a little bit more basic before going to the final one. So as I mentioned, most of the time the comparison is with classical polymers. I'm sure you have, most of the students have studied in classical polymerization. There are two types of mechanism. One is step growth polymerization, and the one is chain growth polymerization. And there was a time initiated by Bertmer and others in 2005 onwards. There is a large number of studies undertaken by them to really find out whether similar mechanistic understanding is possible in these kind of new kind of materials. And it is turned out that similar to the classical polymerization, there are two different types of mechanism existing even in supramolecular polymers. Okay, so I will just quickly go through it. For example, all of you know that it's a step polymerization where each monomer comes with equal reactivity. It's a kind of equal association constant. Similar to the step polymerization, in supramolecular polymerization, you have something called isodesic polymerization. And the other one in classical polymerization, as you know, is a kind of chain growth polymerization, where there is an initiator, there is a propagation, and there is a termination. And similar to the classical polymerization, in non-covalent polymers also, there is something called nucleation growth polymerization or cooperative. So similar to the initiation, we have a nucleation process, and similar to the propagation, we have an elongation process, as you can see that. So this is the nucleation process, and it form long stacks. So it was quite surprising that similar to the classical systems, we have two different types of mechanism existing in supramolecular polymers as well. And it has its own implications, but I don't have time to go through it. But just keep it in mind, most of the time we'll be using nucleation growth mechanisms for structural control and temporal control, because it is often very difficult to deal with isodesic systems similar to the classical system. Okay. So one of the earliest studies which helped us a lot in continuing this frontier areas of research in this field by Chidambar. So he has kind of, he's just recently joined IIT Bombay as kind of assistant professor. So he has done a fantastic, in collaboration with Bala, it's a kind of computational experimental study. He has undertaken a very systematic study on to find out what are the molecular features govern this mechanism of supramolecular polymerization. So the objective was we should be able to design a monomer in such a way that you tell the monomer you follow isodesic mechanism or it should follow a nucleation growth mechanism. This was quite challenging. So we had some proposals kind of we are saying that the presence of long-range interactions in this tag is responsible for such kind of it's important for cooperativity and so on. Okay, so we had later he had come up with experimental systems to really show that our proposal was indeed correct and so on. Although there are exceptions, there are a large number of exceptions in the literature, but at least we use this design principle we learned from this study to design our monomers for future study. I just wanted to say that this is a place we have started a few years back, and still this is a pillar of most of the understanding we are having in this particular field. So then one more thing, because I thought I will slowly go from living to the non-equilibrium to finally to the microstructure, the last problem which I would like to discuss. So if you look into the problem of why we can't have the structural control of supramolecular polymers, one of the issues was, although why we can have a living supramolecular polymers. So as I mentioned, there are chain growth polymerization, but still most of the resulting polymers were polydispersed. We did not have control on their structure or in terms of its dispersity. Most of the time they are highly polydispersed. Okay, so it was again a lot of mechanistic investigation has gone, and one of the challenges people proposed was most of the synthetic system, this nucleation step is extremely fast. That means the activation energy for nucleation step is very low. So also, as a result, what happens if you introduce another batch of monomer to control its length, instead of growing on the existing chain, it will start its own nucleation. Okay, that was quite challenging, because then you don't have any control on the nucleation process. So your structures will be completely polydispersed. You will not have any control on the structure of the polymer. So what was proposed was that you should have a kind of kinetic control in this nucleation process by kind of dormant monomers or something. This was again one of the concepts proposed by many leading groups like Takuzaida and Kazunori. So they have come up with methods to trap these monomers in a kind of dormant state, so that the release of the monomers is not fast into the system, so you can have a control on the nucleation step. Again, I'm not going to the detail, but what you can imagine that they got a kind of complete structural control. So what we were trying to do was, can we have a kind of something similar to the nature? I can see that time is really running out, so I'll wrap up this part. So what we thought that if you look into the nature, this kind of living polymerization and out of equilibrium temporal control are quite ubiquitous. So what nature uses is a very simple strategy. What nature uses is a kind of monomers. For example, if you take it ATP, actin polymerization or microtubule polymerization, but you can see that there is a protein monomers which will not aggregate unless it is triggered by a kind of ATP binding or GTP binding. Okay, so it has a dormant state. You need chemical reactions to convert into an active state, which converts into an assembly. And there is another chemical reaction. For example, in case of actin, it is ATP binding, which assembles, and ATP hydrolysis, which is disassembly. So so it has a perfect control on the temporal aspects as well as structural aspects. Okay, so we try to mimic similar systems. In our case, we came up with, as a chemist, we came up with various chemical fuels like enzymes, chemical reactions, and redox states to really control this structural growth like a chemical reaction-driven self-assembly. Okay, don't worry, this is not the major part of my talk. So the living and transient supramolecular polymerization, we could clearly follow, we could really create by a kind of chemical reaction driven assembly. In this case, it was ATP driven, and the other one was a chemical reaction driven. So basically what I was trying to say that the first two challenges, we have come up with a kind of very bio-inspired method to make kind of chemical reaction driven self-assembly, whereby we can get a controlled growth, thereby structural control we are having, and also we could also make transient material which has a kind of definite lifetime. Some of the early demonstrations like a transient inks recently we published in ACS MI, so that as a material scientist, what one can think about that we can make a system which has a definite lifetime, which is pre-programmed. So you can think about a vesicle, you say that this is a lifetime of a material, so you just burst it after a certain time. So this can avoid the kind of classical stimuli response materials. You don't need another stimuli to break the assembly. Okay, so probably it has interesting functions, which many of the functions we are also exploring in collaboration with many groups. Okay, so my point was, it's indeed the supramolecular polymerization. We are now trying to go to the next level, where we are trying to address next level of challenges like the structural control and temporal control. Can we define the lifetime and so on? So in that aspect, next level of complexity, as I mentioned in the title of the talk, can we control the microstructure of polymers? So if you look into, there are two types of materials I put it. One is block copolymers. All of you know that, which is extensively used in organic photovoltaics. Classical donor acceptor polymers, and then there is, the Vijay Mohan last day talked about heterostructures. So it is a kind of inorganic heterostructures. They are extensively studied. It's between two dissimilar components. We can make this kind of axial inorganic heterostructures. We can make block copolymers. These are all two component systems, which has find a lot of applications in many fields. But what about, can we have a kind of organic heterostructure similar to the inorganic heterostructure? Okay, that means we need to have a kind of two-component supramolecular polymerization, and we should control the organization in such a way that you have segmented organization of one monomer and segmented organization of another monomer. That means you should tell the monomer and you should be able to control your sequence. Okay, so this I'm talking about two component system. It can be even complicated if you take about 3 or 4 component system, just like nature. Okay, and it is going to be really challenge. The reason is, as I told, non-covalent interactions is advantage for controlling the dynamic and adaptive properties. But on the other hand, when you do kind of two-component supramolecular polymerization, one can think that it can have problems. For example, even just take the two-component system, you have 3 different microstructures possible, right? You can have a alternating structure like this, and you can have a kind of blocky structures, which is something would be very useful to make axial heterostructure. And if there is no interaction between the monomers, you can think about they can completely self-solve. Okay, so the question is, can we have a kind of microstructural control so that we can predict all these sequences and we can make it something similar to the axial heterostructures? This was quite challenging, and you know that this is also was quite challenging in the case of block copolymers of conventional synthesis. Just before like synthetic methods like ATRP or ring opening metathesis polymerization came into picture, it was even challenging in the classical polymer chemistry also. Okay, so the question is, can we have synthetic methods where we can selectively make all these 3 sequences in supramolecular polymers as well? Okay. So how to do it? So when we started this program, like a few years back, 2-3 years back, when Aritra started this, the challenges were many. I will just go through the concepts rather than really showing you the results. So we wanted to, as a proof of concept, we wanted to make such structures. Okay, so the first challenge was what kind of monomers we'll take. Okay, so many things has to be taken care. As I told, one of the important thing is monomer exchange dynamics, because if the exchange dynamics is extremely fast, even if you make the blocky structure, it can rearrange and form some other structure. So you should have a good control on the monomer exchange dynamics, as I showed here. That means the monomer goes out of the main backbone and coming out and so on. And how important is intermolecular interactions? Because if suppose this green and red are A and B monomers, and if it is this interaction, AB interactions are much stronger than AA or BB interactions, you will always end up a kind of AB-AB system. You can never make a kind of blocky system. So it is very clear that finite control or delicate balance of intermolecular interactions is also very crucial to get a control of microstructure. Okay, and something which is we also gave importance is like a kind of structural similarity between the monomers, because if they are completely structurally dissimilar systems, always we'll end up a kind of self-sorted system like I shown here. We cannot achieve this kind of copolymers. So as a proof of concept, we were looking for systems which satisfies some of this criteria, and also obviously the mechanism of growth is also important, because even if you look into the classical polymerization, it is most of the time it is a nucleation growth or chain growth polymerization is used to get this kind of multi-component system, because it's often very difficult to play around with isothermic polymerization or step polymerization. So we need to satisfy the other condition that it should also have kind of cooperative polymerization. And obviously the final most important challenge is, unlike classical polymers, again, even if you make a kind of sequence control structures, how to visualize it. Okay, because you cannot use conventional GPC or anything to find out the molecular weight or structure. You cannot do an NMR, because most of the times these things are at much lower concentrations. So you should have some other method. And one of the ways we thought is kind of visualizing them using a kind of super resolution fluorescence microscopy by making use of the fluorescence properties of the system. But again, obviously everyone knows that the resolution is going to be a big issue here, because most of these fluorescence microscopies, we don't have good resolution. So if you really make a microstructure control at the kind of nanometer precision, it's very impossible. It's not possible to even visualize with this kind of fluorescence microscopy. Okay, so keeping this in mind, we have selected a kind of 4 systems, a kind of class of systems. Class of monomers is kind of core substituted naphthalene diimide, which is extensively studied by this Matillet from Subi and so on. So the beauty of this system is you can see that just by keeping the structural similarity and just by changing the core substitution, that means changing few atoms at the core. Core is this this aromatic core I'm talking about. You can change the homolumer gap or its optical properties, electronic properties significantly. You can see that all the way going from oxygen to thiol, it changes from green to red. So we thought, so this is a system whereby minimal structural modification you can cover entire visible spectrum. So that is something which we thought it is very useful to show the concept or to make a microstructure control. So we chose this particular system for our studies. Okay, so we have 2 cases. This is something which we were really interested. Okay, we have 2 different systems, and if you look back to the classical polymerization also, there are 2 ways people made this control on microstructures. One is chain co-polymerization, other one is living polymerization. So chain copolymerization is you take 2 monomers and you just polymerization. So there the sequence is only determined by the reactivity ratio between the monomers. And the other one was kind of living polymerization. That means, as I mentioned earlier, it's a nucleation polymerization, but the chain ends are always active. So as a result, what can happen is at the end of the one monomer polymerization, you can come out with another polymer and it will keep growing. So that way you can have a kind of sequential addition and thereby you can get living polymerization. So thereby also you can get block polymerization. So these were the 2 classical systems. Surprising in classical polymers. Surprisingly, we found that these 2 particular cases we have been dealing with, this belongs to 2 different cases. One is this particular system where we found that this is following almost similar to the classical chain copolymerization, whereas this one is almost similar to the living polymerization. And the reason is, you can see that here, AA interactions and BB interactions are stronger than the AB interactions. So that's the reason you can make such block structures. But in this case, always AB interactions are stronger than AA or BB. So as a result, this can never be formed under thermodynamic control, because always you will get alternating polymers. I hope you guys are following. So if you want to make them, it has to be kind of kinetic control. So this was really surprising for us. All very minute changes. The core is same, the chromophores are same, but slight modifications. You have 2 different types of polymerization mechanisms, and surprisingly, it has a striking similarity between the 2 ways of classical polymerization. So I'll try to quickly go through the results. And so let's take the first case, chain copolymerization. So as I told, this is most of the polymer books you will find it. So if you want to, if the R is equal to infinity, that means reactivity ratio is very high. That means AA polymerization, AA interactions are very high. You will always end up with kind of homopolymerization. But if it is AB is very high, that means AB interactions are very high. That means R values will be much less than 1. You will always end up with kind of alternating system. So what you want is something here, R is slightly greater 1, you will get a kind of block polymer, or R is equal to 1, you will get a kind of statistical polymers. So this is the regime we would be interested. And if you can tune the systems to all extremes, we can get all these sequences. So when it comes to the similar to the supramolecular polymerization, what is reactivity? Reactivity is the intermolecular interactions, free energy of interactions between A and B, because there is no, unlike in classical polymerization, there is no chemical bond forming. So it is basically the free energy of interactions between the 2 systems. Okay. So these are the 2 systems. You can see that this is a thiol substituted derivative. Don't worry about other structural aspects of this polymer monomer, because we have some kind of chiral core, because most of the time we use spectroscopy as a probe to follow the growth of these systems. Okay, so as you can see that spectroscopy, like either absorption or emission or circular dichrosin, can be used to probe the growth, because they are highly sensitive to the way molecules are arranged. Okay, so you can see that this is a thiol substituted system and this is an oxygen substituted system, and this can see that this forms red color fibers and this forms green color fibers. And another beauty is that it has distinct emission features. So even if you make a copolymer, you can separately monitor them, even so you can follow the growth of green monomers and you can even follow the growth of red monomers. So that is what I mentioned, that we can have orthogonal spectroscopic probing. And something which we have done a lot of molecular dynamic simulations with one of my collaborators, Giovanni Pavan from Switzerland, because it is very important to know how, as I mentioned, free energy of interactions, and also how fast is the monomer exchange dynamics. Okay, I'm not complicating the issues. So you can see that the reactivity ratios in this case is 1.17 based on the free energy of interactions. And so you can see that AB is not very strong here. AB strong means we can never form them by putting them together and polymerizing them, as I mentioned before. So you can see that this is 1.17. So it has a good chance that it can form block copolymers. I see that my time is there, all this, but I will take another 5 to 8 minutes, Diksha.
Yes,
yeah?
please.
Yeah, so this is something which is very promising, at least from the calculations. You can see that if you start polymerizing these two monomers, we should end up in a kind of blocky supramolecular polymers. And you can look into the exchange dynamics of monomers. You can see that you can mix two polymers, keep it at room temperature, and you can see that even after a few days, nothing happens. That means exchange dynamics is very slow. So if you make a structure, it will be stable. That's a good sign. And something which we have done to copolymerization, the way we do is we go to high temperature in a particular solvent, mix the both monomers, and cool down very slowly, very slowly in the sense 1K per minute, so that we are forming them under thermodynamic control. And you can probe their growth. This is alpha is the degree of aggregation. That means how much is the monomers in the polymer chain. Okay, and you can independently probe both monomers, and you can see that both the monomers are starting at the same temperature where it starts assembly. Don't forget about the cows. What is more important thing is when we visualize the resulting structures, and this is thanks to Sarit Agasti, who has a wonderful microscope here in our institute. You can see these are really beautiful pictures, which you can see that there are alternating segmented regimes of green and red. At the beginning, we had difficulty to believe it, but then we had an extensive studies, and it was very successful demonstration of a blocky supramolecular polymer just by reactivity ratio. So you just take the monomers, assemble them. Just because of the reactivity ratios, you can control them in a kind of blocky structures. This is very unique, and as I mentioned, these are analogous to the organic axial heterostructures, similar to the inorganic heterostructures. But there was no real way to make such structures. So you can see that there is optical complementarity. So this can be studied for energy transfer. We can couple to some catalysis. So there is basically you can see that self-assembly is a nice bottom-up approach to make this axial organic heterostructures, and we were very happy to see such blocky structures. And we can play around with this length by annealing at different temperatures, because as I mentioned, if you increase the temperature, they will start slightly dynamic. And you can see that you can make really kind of tri-component structures. So you can see this is a green, red, green tri-block structures. We have a good dispersity control. And because the way we do dispersity control is not by GPC, we have to count the number of fibers. That's one of the difficulties in characterizing these systems. But we count like my student counts like 300 fibers of them, and you make a kind of distribution curves like this. And you can see that the polar dispersity indexes are quite narrow, 1.1, which is quite good. And by varying the annealing temperature, you can also see that you can change the degree of polymerization. So this is probably very nice demonstration we can use by clever use of two reactivity ratios of the two monomers. We can indeed make this kind of axial organic heterostructures, which is very unique. Okay, so this gives us a lot of potential to look into these properties like energy migration along the stack, similar to the inorganic heterostructures. So far, this kind of structures could not be studied because there was no way to make such structures. Okay, so I will quickly go to the second. In last 5 minutes, I'll just try to finish the second case. As I mentioned, this was another system. Okay, this was another system, and there was a key difference. In this case, AB interactions were much higher than AA or BB interactions. Okay, so as a result, what happens is if you try to do a similar copolymerization like I showed in the previous slide, always you will get a kind of alternating polymers. You can never get a kind of blocky system, which we would be right now we are more interested in. So definitely we cannot do this polymerization under thermodynamic control. Okay, this is again the system. You can see that beautiful green fibers and red fibers from these kind of two monomers. Again, don't worry about the side chains. These are part of again, this is a cholesterol side chains. This will help in stacking into a kind of one-dimensional polymer. Also, there's a chiral source. We can again use circular dichroism to characterize these systems. So again, you look into the wonderful calculations from Giovanni Pavan, simulations from him, and there you can see that here AB interactions are slightly stronger, and as a result you see that you get a reactivity ratio of 1.03. That means it comes into this regime. So if you try to do this polymerization under thermodynamic conditions, you get completely statistical polymers. You can never get a blocky system. Okay, so that is what exactly we have observed. So you mix the monomers, you heat it to high temperature, you bring down, and you can see that you can probe both the system, and forget about the spectroscopy. But if you look into the microscopy, this is the merged channel, this is probing to the green channel, this is probing through the red channel, and you can see that both the monomers are randomly arranged in the polymer chains. As expected, the reactivity ratio plays a crucial role. So in this case, how do you make a block of polymer? So it's very clear that thermodynamic conditions we cannot make it. So then that is why, similar to the classical living polymerization like ATRP, which Machersky developed, we need to have a living polymerization. That means in supramolecular chemistry, it has to be under kinetic control. What you do is you make a particular seed, one of the polymer polymerize it fast, one of the monomer polymerize it fast, and use the other monomer introduce at a later stage. And if the ends are living, you will end up in growing the second part of the monomers. So that is why we can, as I shown here in the schematic, you have to make one polymer, then you have to introduce another polymer into another monomer into it. You should do. But it is not easy, because there is a possibility that this second monomer can have its own nucleation properties. So it's not necessary that it grows on existing chain. That's why we need a clever design, as I mentioned. We need to trap these green monomers in a kind of dormant state, and then you introduce into the this seed, just like a classical synthesis of inorganic heterostructures. And if this green monomer is in a kind of dormant state, it will not start, once it starts growing, it will not start its own nucleation. On the other hand, it will start growing on the existing chain. So again, how do we make the dormant state is, since the time is not there, I'm not going to the details, but if you have questions, you can ask during the question answer session. So we are able to make the dormant state, and the critical experiment is here. Okay, so you make the first polymer, which is kind of the green color, and you introduce the other red monomers in a kind of dormant state into the system. Okay, and this is the dormant state, it cannot start its own nucleation, but as soon as you introduce into the seed, heterogeneous nucleation happens and it starts growing into the system. So this it's again a spectroscopic probing. We can selectively probe the growth of the red monomers in the system. So it starts, you can see that as soon as you introduce the seed into the system, it's immediately starts growing. So if you don't introduce the seed, the monomer remains in a kind of dormant state. So as a result, one can imagine that this is a kind of kinetic control, because if it is under thermodynamic control, it can never form a kind of block system. It will always form AB-AB system. But by a kind of kinetic control, we can really make kind of blocky systems. As you can see, although imaging is not great, so we have enough proof that we are indeed forming kind of blocky structures. So these both cases we have extensively characterized. You can see that the supporting information will have kind of 50 pages with an extensive collaboration, because the systems were highly complex. So we had good collaborations with molecular dynamic simulation guys. We had collaborations with imaging people. We have collaboration with functional people to really look into the functional aspects of this. Okay, so we have, you can see that MT simulations also supports that indeed it forms a blocky chains. I'm just trying to give the concepts. Okay, so where do we go from here? Last 2 minutes, I'll try to summarize the results. So I told that we can make this kinetic control and thermodynamic control. We can make this new kind of microstructure in this kind of supramolecular polymers. So we need to really get a kind of controlled degree of polymerization dispersity. That is, we are trying with some other systems, because in the previous case the imaging was not great. This is a new system we are getting developed. So you can see that you have a kind of polydispersity values remarkable, similar to the ATRP system, like 1.06 ideal. So it's very close to a kind of monodisperse system. So we are very happy that this strategy works with other systems. Obviously, two-component system is not the end of the world. We need to go more and more complicated system. Can we have a three-component system? So can we have a kind of tri-component multi-block structures? That's something which we are trying. We are now introducing a kind of blue system into it. And you can see that we have blue, green, red. The challenge is to visualize them. And if you believe me, we have tri-block systems here. We are trying to improve the imaging aspects here as well. We are trying to see, because in nature, if you look into this, what regulates this monomer sequence? It's by kind of externally programmed. It's not like in this previous cases, you might ask me, oh, you are physically adding it, okay? So that way we can never reach the complexity what nature is having. So we should have a kind of external stimuli. What we are doing in this system is like a kind of pH control. At least you modulate the pH and you define a pH, and that time another monomer comes and start stacking. This is in collaboration with one of the German group. So we are now trying to this kind of peptide segments which are known for pH responsive assemblies. So we can have a kind of controlled pH variations whereby these molecules start assembling on top of each other. So this is something which is going on. And the most important part, why we make it, okay? So as I told, these are very unique structures. Although these were kind of inorganic heterostructures are well studied for many decades, organic heterostructures are pretty new. So and similarly, whatever we did it with inorganic heterostructures like catalysis, exciton transport, everything can be done with organic heterostructures. So preliminary studies in collaboration with Arindam from IIT Bombay. So we are now trying to look into, because this is kind of energy donor and acceptor axial organic heterostructures, and we have very interesting results because there is a large exciton migration we have seen, and there are preliminary results we are further looking into it. So these are kind of light harvesting organic heterostructures we can make it out of it. So that brings to summary. Sorry that I took 10 minutes extra, Diksha. So this is so what we are trying to do is to have a kind of structural control. We were trying to have a temporal control, and finally, very recently, we are now moving one step ahead where we are trying to bring a kind of microstructural control. That means if you have more than one monomer, can we really define the positions of this monomer in the chain? Obviously, there are challenges. We have to go for more complexity, and I think we are slowly progressing, and we are very interested in exploring its functions because these are ideal structures. We can look into coupled catalysis and so on. And we are also looking into higher order structures because in inorganic structures, people talk about layered heterostructures, radial heterostructures like graphene, MOS2, and so on. So we are also in the process of making 2D organic heterostructures. It's quite challenging. We have some preliminary results, but we are in the process of going on. I should thank my collaborations. Molecular dynamic simulation is an extensive, because this works, especially the microstructure part is highly complex. We need a lot of expertise. So MD studies were very crucial. So Bala from JNC, my colleague, and also Giovanni Pervin, they helped us a lot in understanding the monomer exchange dynamics and free energy calculations. Sarita Gassi has a wonderful, I did not mention, most of the images were SIM images, that is structured illumination microscopy images. So that is done in Saris lab, and Arindam is exploring the various functions of these unique organic heterostructures. With that, I'd like to stop, and most of the results are most of the multi-component structures are done by Aritra, who is a final year student, and yeah, so, and also I'd like to thank Professor Rao. So we have a kind of, I belong to this new chemistry unit, which is a brainchild of Professor Sena Rao, where we work at the interdisciplinary areas and so on. I'd like to thank all the funding agencies, and thank you for your attention, and I'll be looking into the questions now, right?